I am Aarote, Junior College Teacher, RBNB College, Rampur. Subject Biology. Today we see the chapter that is the Enhancement in Food Production. Previous lecture we see the role of microbes in biofertilizers. For the production of biofertilizers, so many microbes are used for the formation of biofertilizer, because when biofertilizer is produced by the microorganism, they are very helpful to increase the soil fertility and also to decrease the pollution of the environment and increase the productivity of agriculture and a benefit to the farmers. So suppose we see near about into the environment, 72% nitrogen are present into 78% nitrogen are present into the free atmosphere, but the plants have not ability to fix this free atmospheric nitrogen. If this free atmospheric nitrogen is fixed by the plants, that is, we have no need to purchase the chemical, organic as well as chemical fertilizers. But these are the bad thing to the farmers as well as our ecosystem or as environment. We have need to purchase the biofertilizer as well as to purchase the chemical fertilizers. So some microbes, near about 72% of the nitrogen are present into the atmosphere, but plant have not ability to fix it, because why? These are the some affinity to fix this nitrogen from the environment. It is cannot be found inside the plants, that is higher plants, but this type of the nitrogen will be fixed by the some microorganisms. So these microorganisms plays an important role to fix the atmospheric nitrogen. Suppose previous lecture we see the role of microbes is a biofertilizer. It was near about the 5 types. First was the nitrogen fixing biofertilizer, second was the phosphate solubilizing biofertilizers, then compost making biofertilizers, cyanobacteria as a biofertilizers, fungal biofertilizers. It was the 2 types, that is ecto and endomycorrhiza. But now today we see that is the next type of it, that is the biofertilizers as a microorganism. What is biofertilizer? What is the microorganism? That is the microorganism plays an important role to fix the atmospheric nitrogen and to provide or as a supply to the plants. Between it, first biofertilizer is the Rhizobium. First biofertilizer is the Rhizobium. What are the Rhizobium? Rhizobium are the rod-shaped. Rhizobium are the rod-shaped. Rhizobium are rod-shaped, non-spore forming nitrogen fixing bacteria. These are the gram-negative. The bacteria are the gram-negative, non-spore forming and nitrogen fixing bacteria. It plays an important role. These bacteria have ability to fix the atmospheric nitrogen, because why? They contains, that is these bacteria contains the NOD gene and NIF gene. These bacteria contain NOD gene and NIF gene. What is NOD gene and what is NIF gene? NOD means that is these bacteria will be found into the nodule of the dicotyledonous plants. NIF means that is the fixation of nitrogen from the atmosphere. So these both, that is 2 type of the genes will be found, that is NOD gene and NIF gene. NOD means that is these will be found into the nodule of the dicotyledonous plants. NIF gene means that is nitrogen fixing gene are present in this biofertilizer, that is the Rhizobium. They form a symbiotic association with the roots of leguminous plants. They form the symbiotic association. What is symbiotics? That is cannot both having the benefit but not have a harm to the this Rhizobium as well as leguminous plants. So these are the symbiotic association of the leguminous plants, and these have ability to fix the atmospheric nitrogen. So nodule formation on, that is a nodule formation on the root, it have ability to multiply inside the nodules. These Rhizobium have ability to multiply inside the nodules and have ability again to fix the atmospheric nitrogen into the organic forms. When this rhizobium is come in the contact with the leguminous plants, they form the nodules. When after the formation of these nodules, they increase, or as a division of these nodules will be seen and have ability to fix the atmospheric nitrogen, and thus it is used by the plants as a nutrients. It is used by the plants as a nutrient. That is example for these are the best example: Rhizobium leguminosus. Rhizobium leguminosus is the best example for pea plant. Is the best example for the pea plant, and Rhizobium phaseoli are the best example for the different type of the beans. So these two type of the rhizobium are used for the fixation of nitrogen. One is the Rhizobium leguminous, only for the pea plants, and other is the Rhizobium phaseoli have ability to fix the all type of the beans nitrogen into the all type of the beans. So what is the structure of this rhizobium? Suppose we see the structure of this rhizobium on the root nodules. These are the dicotyledonous plants having the number of roots are there. On these number of roots, so many nodules will be produced on it. Number of nodules will be produced. It is known as the these are the number of nodules are produced on these roots. Suppose we see the structure of the single nodules. If we take a tiers of the nodules, tiers of nodules will be seen like of these. These are the nodules. These are the cellular structure. These are the cellular structure of the, if take a tiers of root nodules. These are the outer wall. Then here again, suppose we see the number of rhizobium colony can be found here. Number of rhizobium colony will be found here. The central part, they contain the conductive tissue. Central part contains the conducting tissue. Then these are the cellular structures between these roots. So upper part, these are the root nodules. These root nodules contains the bacterial colonies. Here are the bacterial colonies. Here are the bacterial colonies. Then this is the root. These are the xylem, and surrounding to it these are the phloem. Suppose see the structure of root nodules, that is tiers of root nodules. It shows like of this. These are the root nodules will be developed on the roots. So they contain the bacterial colonies. They have ability to fix the atmospheric nitrogen, and this nitrogen directly takes by the dicotyledonous plants or as a leguminous plant. Next we see that is the azotobacter. Next microfertilizers or as a biofertilizers. Next microfertilizer. Or as a biofertilizer is a azotobacter. What is the azotobacter? It is an imported and well-known free-living nitrogen-fixing bacterium. These are the well-known. It is the well-known free-living, that is free-living into the atmosphere, and these are the nitrogen-fixing aerobic and non-photosynthetic bacterium. These are the aerobic and non-photosynthetic bacterium have ability to fix the atmospheric nitrogen. So this bacterium is associated with, that is, what is the association of this bacteria? This bacterium is associated with the roots of grasses and some higher plants. Or as a, suppose we see that is these are the microorganism, that is Azotobacter. It is an important and well known for the free living nitrogen fixing aerobic, non-photosynthetic, non-nodule forming bacterium. This bacterium is associated with the root of some grasses, and some plants. That is the grasses. These are the monocotyledonous plants, and they have used. It is used the biofertilizers for all. These are the biofertilizers are used for all non-leguminous plants, especially that is the rice, cotton, and vegetables. These Azotobacter are the biofertilizer. What is the, if we see the nature of these biofertilizers are free living, nitrogen fixing, aerobic, non-photosynthetic, that is the photosynthesis cannot be found between it. So these are the non-photosynthetic, non-nodulated, nodules cannot be seen on it. So non-nodulated, these are the bacterium. They will be found or seen into the roots of grasses as well as plants. So these type of the Azotobacter bacterium have not ability to fix the nitrogen with the leguminous plants, but the nitrogen get fixed by these plants only, that is like of the rice, cotton, and some of the vegetables. Then suppose we see the next type of the microorganisms as a biofertilizer. That is the third type, the Azospirillum. Azospirillum these are the third microorganism, that is the Azospirillum. These are the free aerobic, these are the free living aerobic nitrogen fixing bacterium associated with the roots of corn, wheat, jawar, that is the monocotyledonous plant, that is Azospirillum. These are the living bacterium associated with the corn, wheat, jawar. It fixes the considerable quantity of nitrogen, is near about 20 to 40 considerable nitrogen fixation is near about 20 to 40 kg per hectare. Per hectare, these are the, that is these near about 20 to 40 kilogram nitrogen will be fixed by these plants like of the corn, wheat, jawar. The Azospirillum have ability to fix the nitrogen in the field like of the corn, wheat, jawar. They have ability to fix near about 20 to 40 kg nitrogen per hectare, and it means that is leguminous plants such as cereals and millets, that is cotton seeds, have also ability to fix this type of the nitrogen. Some leguminous plants which are this oil seed or as a cotton or as some cereals and millets, that is kadadhanya vagare, cereals and millets have ability to fix the atmospheric nitrogen by the Azospirillum. So the very great formation of nitrogen can be found by this Azospirillum in this field. Near about 20 to 40 kg nitrogen will be synthesized by this Azospirillum in this farm or as a field. The next as an important, that is Anabaena. These again as I found very important microorganisms for the formation of the biofertilizer. Next, that is fourth type, that is Anabaena. What is the Anabaena? Anabaena is nothing but the genus multicellular. These are the genus multicellular cyanobacteria. These are the multicellular cyanobacteria. Suppose see the structure like of these, having the beaded like structure. Number of beads are arranged ones to the others. All cells are alike, but only some cells are different from each other. It is known as the heterocyst. This heterocyst is different than the other cells. The size of this heterocyst is larger than the other bead. These heterocysts have ability to fix the atmospheric nitrogen. These are the structure of Anabaena. These are the cyanobacteria, that is multicellular cyanobacteria, having the beaded like structure. A single cell between it. These are the heterocysts. Because these cells are large in size than the other cells, they have ability to fix the atmospheric nitrogen. So these Anabaena can be found into the field like that is the coralloid root of cycas, or as some paddy fields the Anabaena and nostoc can be found into the paddy field as like of the where the water is stored abundantly, and also they will be found into the coralloid roots of the cycas. Coralloid root of cycas. These Anabaena can be found again with the, that is coralloid root of the cycas, and the plants like of the Anthoceros thallus. Anthoceros thallus. Again, these Anabaena found into these like of the plants like of the paddy field as a paddy field, as well as coralloid root of cycas. Anthoceros. They will be found into the colonies of these cells, and probably these cells are again as like of the beaded like structure. Long and elongated cells can be found, but between these, that is one cell is a other that is different than other cells, that is these are the large in size. They have ability to fix the nitrogen because it is known as the heterocyst. The next part of this, that is a microorganism. Next organism to fix the biofertilizers. These are the Azola. Next, microorganisms to fix the to produce the biofertilizers. It is known as the Azola. The Azola is a free floating plants. The Azola are the free floating water form. Azola are the free floating. Free floating form. If we see the structure of this Azola, it looks like as these are the free floating. Leaves are added once to the above. These are the root portion. They will be free floating above the water. I have ability to float on the surface of water. Suppose we see the Azola. Azola plant consists of the free floating rhizome. The Azola plant consists of the free floating rhizome. It is also known as the root. It is also known as the root and rhizome. They are free floating on the water. If we see the leaf of these, they are lapping bilobed. The leaves are lapping and they are the bilobed, that is dorsal and ventral ribs can be seen on these plants, or as a leaf. Dorsal and ventral leaves can be seen on it, or as a lobes can be found on the dorsal as well as ventral surface. The dorsal lobe, that is Anabaena filaments are present in the on the dorsal lobe. The Anabaena filaments can be found in the parenchyma. Parenchyma, these are the tissue found in these plants. They are helpful for the floating these plants above the water. That is parenchyma. It is also known as the cavity of the air. This cavity helpful for to float these plants above the surface of water, and surrounding to these plants, that is colony of nostoc can be found. These nostoc will be seen, or that is the Azola can be seen, the colony to the dorsal surface of the Azola plants, and how they produce the colony to fix the atmospheric nitrogen to the dorsal side of these Azola plants. Azola also found into the rice field have ability to synthesize or secrete the biofertilizer but important role between these, suppose we see the important role for this Azola have ability to fix the atmospheric nitrogen, because why? These Azola, these are the fern will be found into the free floating, but they present inside the plant. So the nostoc and Anabaena can be found inside the water. So they have ability to fix the nitrogen to this dorsal surface of the free floating Azola. Because why? The parenchyma are present inside this plant. It is known as the cavity. The cavity have ability to float the plant on the surface of water, and surrounding to it, that is the colony of this nostoc can be found. Some nostoc will be found into the rice field where the water are present abundantly. There the colonies of nostocs can be found. They have ability again to fix the atmospheric nitrogen. If we see the LS of this leaf, if we see the LS of this Azola leaf, it can be found. If we see the Azola of leaf, here are the parenchyma. These are the air space, surrounding to air space. These are the cell. Here these are the photosynthetic cell. These are the photosynthetic cell. Here are the air cavity. It is nothing but the parenchyma, cell of the parenchyma to float it, and surrounding to it, the colonies of nostocs can be produced. The colonies of nostocs are produced here. Probably the colonies of the nostocs will be produced here, surrounding to it. And in between this colony, here the Anabaena can be found. The Anabaena are found into the cavity of air. It is also known the parenchyma. So these are the central part. They contain the Anabaena. They fix the atmospheric nitrogen. So nowadays in our country, many biofertilizers are available. Nowadays in our country, many biofertilizers are available in a market. To reduce the use of chemical fertilizers, to reduce the use of chemical fertilizers, and also reduce the pollution. So biofertilizers plays an important role for the that is the increase the productivity of the agriculture crop, as well as to reduce the pollution. If we see some benefits of the biofertilizers, what are the benefits of the biofertilizer? Some benefits are there, because biofertilizers is non-polluting and less expenditure, very easily available into the market. This type of the biofertilizers very easily available into the market, because these are the less pollutant, have ability to increase the agriculture production, and very efficient to the farmers, because to decrease the cost. Suppose we see first use of it, that is the low cost, and it can be used as a marginal farmers having the less cost, because very easily they are available into the markets and having the low cost to the marginals of the farmers. These are the first use, or as a first benefit of it. Second, we see these are the microorganisms, or as a biofertilizer, free from pollution hazards. That is, these are the free from pollution hazards. If we use the chemical fertilizers can pollute air, water, soil. The chemical fertilizers can produce the air, water and soil, but these biofertilizers cannot pollute the air, water as well as soil. So these are the free for the pollution. Then next, as an important factor, they have ability to increase the fertility, soil fertility, because nowadays by using the number of chemical fertilizers, that is if we use the chemical fertilizers, the fertility of soil will be decreases. So if we using these biofertilizers, the biofertilizers have ability to increase the fertility of the soil. Then BGA, that is blue-green algae, as a biofertilizer secretes the growth promoting substances, organic acids, proteins and vitamins. BGA these are the blue-green algae also found in, these are the microorganism or the biofertilizer. They have again has the ability to secrete the growth of the plants, to promote for the substances. Then organic acids, proteins and vitamins are produced by these busy or as a blue-green algae. Then next important, that is azotobacter. These are again as a biofertilizers or as a microfertilizers, microorganism having the biofertilizers, azotobacter. They supply the nitrogens and antibiotics in the soils. That is, azotobacter have ability to supply the nitrogen and antibiotics in the soils. As an important benefit to the soils, that is, the biofertilizers increases the physico-chemical properties of the soil, like texture, structure, pH, water holding capacity of soil, and providing nutrients and organic matters to the soil. So these are as an important factors, that is, the biofertilizers have ability to increase the physico-chemical properties of soil. What is the physico-chemical properties of soil? Like of the texture, their structure, their pH of the soil, water holding capacity of the soil, and to providing the nutrient and organic matters to the soil. So biofertilizers, these are the that is, microbes plays an important role for the production of biofertilizers to the soil. So farmers have need to go toward the micronutrients as a biofertilizers, because why? Less cost, then that is the increases of the soil fertility, and pollution free environment will be produced by the microorganism. So we suggest to avoid the chemical fertilizers and to use the biofertilizers to eco-friendly to the farmers as well as ecosystem. Now today these topics will be end or as over. Next time we see the next topic. Thanks.